Hello, good afternoon. Thank you for being on time. We are going to get started right away. And today we will be learning lecture 3, which is about cells. So let's get right into it. Start with some introductory information. You might remember this picture from earlier on in the course where we were talking about characteristics of living things. And one of the characteristics that we talked about is that there are complexity and organization associated with living things. And I show you this hierarchy of organization. Lecture 2, we spent a lot of time talking about the macromolecules, the four main type, which you actually will be quizzed on next week. And so now the next thing up the chain here is organelle and cells. So that's going to be the focus for pretty much the majority of the rest of the course, all the way until lecture number 9. And then in lecture 10, we're going to move on to tissue, which will transition nicely into organs and organ system for next semester. So today we will be talking about what a cell is, and we will learn some of the organelles that exist inside the cell. So how big is a cell? Well, cell ranges between 10 micrometer to 100 micrometer, depending on what kind of cell you're looking at. Some cells are bigger than others. Plant cells might be bigger than animal cells, for example. And in case you forgot, 1 meter is equivalent to 1,000,000 micrometer. Okay, so that's the range for measurement for cells. And then if we look inside the cell, there are many structures which we call organelles. Right? So a mitochondria is an example of an organelle, and those are about 1 micrometer, similar size to bacteria. And if we go even smaller on the scale, things like viruses, they are measured in nanometers. Okay, nanometers. So 1 meter is 1,000,000,000 nanometers. Okay, so this gives you a perspective as to how big various things are. Okay, so some of these things we're able to see with our naked eye, but if we want to have a detailed look at the cells, then we would need the help of a microscope. So in this picture, in this slide rather, we have 3 different cell types here, and they all look kind of different. Okay. Excuse me. Some cells are spherical and they like to connect with each other. And these are actually bacterial cells. We call them Streptococcus in this particular case. You might have heard of them before. They can cause things like strep throat. This over here is an amoeba, which are found, which are found in fresh water. Okay? Sometimes they can make their way into swimming pools, and they could cause people to become ill. What about the things down here? Anybody have an idea of what that might be? Any guess? You might have seen this before. Is in our body somewhere. No guess? Okay, so these are neurons, right? Which you will find them in the brain, for example. So what exactly is a cell? Well, a cell is defined as the smallest unit that has the properties of life. Remember all those 7 characteristics that we talked about? Complexity, ability to maintain homeostasis, respond to stimulus. So all these things are still true on a cellular level. If you go any smaller than a cell, if you go inside of a cell, then the stuff in there no longer retain these properties. Right? And so they're not really alive anymore. So cell is basically defined as the smallest unit of life. Despite the fact that they look very different, all cells have common features that are shared. For example, all cells have a cell membrane. So here is a cell membrane. It's kind of like the boundary of a cell. You can think of it like that. And inside the cell membrane, it's filled with almost like a fluid / gel-like substance, and we call that the cytoplasm. Okay. Another name for cell membrane is plasma membrane. They mean the same thing. And in addition to the cell membrane and the cytoplasm, you will have some genetic material. Okay, genetic material, right? Such as DNA. Okay. So regardless of what kind of cell you're talking about, right? It doesn't matter if it's a bacteria, if it's a human cell, plant cell, amoeba, all of them are going to have these 3 features. Okay. Now I'm going to talk about the cell theory. Okay? The cell theory has 4 parts to it. First part of the cell theory states that every living organism consists of 1 or more cells. If it has 1 cell, then we say they are unicellular. Unicellular. They have more than 1 cell, and we say they are multicellular. So over here, we have something called the paramecium. It's in the same category as the amoeba that I showed you before. They tend to live in fresh water, and it's only 1 cell. Right? This entire thing is an organism, and it's just 1 cell. Okay? This here is called a C. elegans. It's like a type of worm, microscopic worm, worm-like organism, and it's about 3,000 cells. Right? This is what people call like a model organism. Okay. Model organisms are organisms that scientists knows a lot about, and then they do research on them to test for various things. Okay, like a new drug or genetic manipulation. Right. So C. elegans is an example of a model organism. Fruit flies is another example of a model organism, and so are like mice. Okay. And humans, we have trillions of cells. Trillions of cells. Of course, as you grow, you get more cells up to a point. Right? If you're an adult, then the number of cells you have stays the same. Right? As new ones are made, old ones die off. So, it's some kind of equilibrium. But, for a child, the number of cells keeps on increasing as they grow to adulthood. So first part of the cell theory, every living organism consists of 1 or more cells. Second part of the cell theory goes like this. A cell is the smallest unit of life, which is what I told you at the beginning, right? It retains all the properties of life, and they are actually individually alive as part of a multicellular organism in some cases. So each of our trillions of cells, they are individually alive. In fact, you can take our cells out of our body, and then you can grow them in a petri dish. Okay, that's what these things are. Okay, they're called petri dish, and you can give them nutrients. Here, I'll write down for you in case you're interested. Petri dish. The red solution is like nutrients that you can feed the cells with, and they would be quite happy for some time. You'll be able to grow them for a period of time, and then eventually they will die, just like they would in our body, right? Okay. Now there was a woman. Her name. Yep. Go ahead.
I know it's not the.
That's okay.
Yeah, but so you mean that if you take a whole organism, it can live. Let's say the heart or the lung or those things.
Sorry, you can take the heart and the lungs of someone, and then what?
And if we put outside the body, if we put them in a petri, they can live.
Yeah, so like we're not talking about the whole organ, right? But like if you take cells from a person, like if you take their liver cells or something, then you would be able to grow the cells in the petri dish for some time. Okay. For the entire organ, you can keep them alive if you put it on ice. That's actually what happens when people do organ transplant, right? When a person pass away and then they agree to donate their organ, they actually have to keep the organ fresh by putting it on ice. And you can keep it that way and alive for some a short period of window before you transplant it into another person, right? But when we talking about cells, the period you can keep them alive is much longer than the entire organ. Okay.
Okay.
Yeah. So there was this woman. Her name was Henrietta Lacks, and in the 1950 ish, she was diagnosed with cervical cancer. Right? And what the scientists and the doctors at the time was doing was they took a biopsy, they took a sample of her tumor, and they tried to study the tumor cells in a dish, okay, in a petri dish. Okay, and what they found remarkable was that regular cells, you can grow them for some time and then they die off. That's what I told you, right? But these cancer cells, they were able to grow them continuously. Okay, like when they fill up the whole dish, they could take it and then separate them and then put it into like 4 other dishes, and then they would continue to grow. And it seems to be growing and dividing infinitely. Okay, eventually, they created what's called the HeLa cells, named after the woman's name, right? Henrietta Lacks. And they use these cells to study a bunch of things, including using them to create vaccines, right? To study treatments for things like tuberculosis, Ebola, HIV, and cancer treatments, right? And so these cells are very different nowadays than the original batch, but they nevertheless came from those cervical cancer cells that were taken from Henrietta Lacks. Okay, in fact, you can buy them online, right? If you want to buy some HeLa cell lines, 20 microliter is gonna cost you $486. Right? This is an old slide. It's probably more expensive nowadays. Okay, I looked this up like a year ago. But it's a very small amount, but it contains a lot of cells in the sample. And one of the reasons why it's so expensive is like, people who buy these things are usually scientists who are doing research, right? And once you bought them, you can grow them yourself. In fact, I've done it before in my graduate work. We got this cancer cell line and we grow them in the dish. And once you have a full plate, you can actually take some and put it into little jars and we freeze them at minus 80 degrees. Okay? So when we need to get more cells, we just thaw it from the freezer and then we grow more. Okay, and then we freeze some back. Right? So you can kind of keep on using them for a very, very long time. Okay. Do these cells reproduce asexually? Yeah, that's a great question. Yeah, we're going to talk more about that later. Short answer is yes. Okay. Once you give them the right condition, they would just divide and divide and divide and make more copies of themselves. Okay. So that's a great question, actually. Even though we are sexual being, in the sense that we reproduce with a partner, our cells reproduce asexually. Okay. Which brings us to the next point. But before the next point, one more slide on the story here. So the company who sells these HeLa cells, they are making a lot of money, right? A lot of profits from selling these to researchers, to people to study the cells, and so the woman died from her cancer, but her estate and her family actually sue the company Thermo Fisher over the HeLa cell lines. Okay? They're seeking for compensation, saying that they did not have her consent to basically commercialize her cells. okay? And the cells are being cloned continuously to turn a profit. Okay, so the company tried to, dismiss their case. There was a bit of back and forth, but ultimately, the family won, and compensation were paid to the family regarding the HeLa cells. Okay, you can watch more about, you can read more about these things by searching it online. Okay, so that brings us to the next part of the cell theory. How do you get new cells? Okay, turns out all living cells come from division of pre-existing cell. Okay, here is a picture of something. Anybody want to guess what this is? I think people don't like to guess things. I don't know why. What do you think you're looking at here? It's not an animal, I'll tell you. Okay. What do you think you're looking at here?
Plant.
Yes, it's plant. What kind of plant? You probably ate this before. Anyone?
I don't know. I forgot.
Yeah. Okay. Yeah. Well, this is showing the process of mitosis. Good. But this is onion, guys. Okay, that's what onion looks like when you look at it under the microscope. I mean, it's not pink. You can stain them with different dyes to highlight certain features, but that's what onions look like. This is one onion cell, and this is an onion cell that is dividing. Okay? These things that you are seeing here, those are actually the DNA, the genetic material. Okay, so all cells come from division of pre-existing cell. Right? You and I, we both started as a single cell. Okay, sperm and egg came together and it created the first cell, and that keeps on dividing and dividing and dividing, and eventually it became you. Right? It became me. Right? So that's one cell, and the cell is going to divide and create two genetically identical cells. This process is called mitosis. And we're going to learn a great deal about mitosis in lecture 7. Next part of the cell theory states that cells contain hereditary material which are passed on during division. Does anybody remember how many chromosomes you have in your cell? Anyone?
23 sets.
23 pairs, right? 23
23
pairs.
sets.
Yeah,
Yeah, 46.
so 46 chromosome. So every time your cell divides, you actually have to copy all the genetic material, and each of the new cell that you create will also have 46 chromosomes. Okay, you have to double the genetic material before the cell divides. Otherwise, you start with 46 and you divide, you get 23, 23. If you divide again, then it becomes what, like 11.5, right? Like doesn't make sense that way, right? So we have to double them first. Okay, so those are the 4 parts of the cell theory. Now, cells are very small. You cannot see them with your naked eye, and so we need to have the microscope to help us do that. And the first microscope that was documented was created by this Dutch scientist named Antony van Leeuwenhoek. That's how you pronounce his name, Leeuwenhoek. He wasn't actually a science by training. I think he was a tailor selling, clothing and fabrics. But as a side interest, he liked to make lenses. Right? And one time he made this very small lens, and then he was just looking at water through the lens, and he noticed there is an entire world that is unseen in the water when you look through the lens. Right? He saw all these things moving around. Some have tails, some are dashing around purposefully. So he called these things that he saw animalcules. Okay, they are molecules that looks like animal, right? So in fact, the things like amoeba and paramecium, that's the kind of stuff that he was able to see in the water. sample that he gathered. Okay, no one actually believed him. Okay, he made a detailed catalog of all the things he saw. He drew some pictures of it, and then he showed it to people, and people think he was just like delusional, making all this stuff up, because other people weren't able to replicate the result because they didn't have the skill to make the kind of lens that he did. Okay, so he invited people with like authority, I guess, right? Like he invited people who are important figures in society to come and look at what he saw through his microscope, right? Invited like politicians, lawyers, and those people eventually vouched for him, and then that created like a whole field of microscopy. Yeah, so that this device here was the microscope that he created. Nowadays, we can get microscope very easily. Okay, you can buy one from Amazon if you want. The ones we use at school are these light microscope. They're not too expensive, like about $200-$300 each, and they could magnify up to 1,000 times. Okay, magnify up to 1,000 times, more than enough to look at cell samples and other things. The really expensive microscopes can magnify up to 2.5 million times. Okay, they are called electron microscope. That's what the EM stands for. Right? These are light microscope, uses light to observe what you're trying to see. These uses beams of electrons. Okay? And depending on whether you use scanning electron microscope or transmission electron microscope, you will be able to see different things. For the SEM, you will be able to see a three-dimensional structure of the specimen, whereas the TEM will show you a cross section of your specimen. Okay, it really depends on what your application is, and you will use different methods. Now these microscopes are like very expensive. Okay, easily between 1.5 to 2.5 million dollars for the whole setup, software and everything. Not something typical school can afford. Some universities might have it. Research facilities will definitely have them, and even maybe at the hospital. Okay. Anybody know? I know I'm asking you to guess again, but these things, what is it? Is in the body. Okay, somewhere in the body, in the upper half of the body. Okay. Anybody want to guess what that is?
2 places, maybe.
What is it? I'm so sorry.
I said 2 places.
2 places.
Yeah, lung and intestine.
Okay, you're very close. Okay, very close. These are actually in the airway. Okay, in the trachea, in the airway, and they are little hair that will help sweep up like dust particles and pollutants that we breathe in, and then sweep them up to the throat where you can spit it out or you can blow it out of your nose. Okay, we'll learn more about those later in the lecture. People who smoke, heavy smokers, they damage these structures, and they have no way of getting rid of stuff that are stuck in their airway. Right? So over time, they develop the smoker's cough. Okay? They have to cough it out. Right? And then you see smokers, they spit a lot. It's because of the stuff that they're coughing up. They got to get rid of it, right? So that's what it looks like in the airway. All right, let's do a poll question. Go ahead. Right, most people got it right. The correct answer is D. A cell is the basic structural and functional unit of living organisms. Let's do one more. All of the following are present in all cells except for what? Okay, looks like people are having a bit of trouble with this one. Let me just go back quickly to show you. Previously, I was telling you that common features of all cells. We have the cell membrane, the genetic material, and the cytoplasm. This question is asking you for the exception. Okay, so the exception is nucleus. Not all cells have a nucleus. We haven't talked about the nucleus yet, but we will. Okay, but not all cells are going to have the nucleus. Now, while I have most of you here, I want to bring your attention to the course homepage, okay? If you log on to eCentennial, I posted the concept check question answers for lecture 1 and 2. Remember last class, we didn't have enough time to take up all the questions for the proteins and for the DNA. So the answers are here, okay? Please download it and take a look at yourself. Take a look at it yourself. The quiz once again will be on Monday, and this is the quiz link. Okay, you just click on it, and it will take you directly to the quiz. If you haven't already done so, you need to install the Lockdown browser. If you go back to like earlier announcement, it will tell you how to do that. Okay. Make sure you do the practice quiz to help you study a little bit, but more importantly, do the study guides and go through your lecture notes. There will be 2 short answer questions, 17 multiple choice, and 5 multi-select questions. Multi-select questions are questions that have multiple correct answers. It might have 1 correct answer, 2, 3, 4, 5. Doesn't matter. It depends on the question. You just choose as many correct one as you need. Time limit is 45 minutes. Someone asked we could make a cheat sheet. Yeah, you can make a cheat sheet. Okay. In fact, I encourage you to make a cheat sheet, help sheet, whatever you want to call it. That would help you along with the quiz. Okay. If you require testing accommodation, you got to book an appointment with the Center for Accessible Learning and Counseling Services and send me the documents before the first quiz. If you don't know what I'm talking about, then you can also click on the link and see if it fits your criteria. Where can you find the concept check? Right here. It's the first announcement. Okay. You just download the file and then all the answers are gonna be there. Okay. Sheila, did you have a question? I see that you're mute.
Oh, sorry, that was a mistake.
Okay, no problem.
Sorry.
Yeah, that's okay. All right. We do have class on Monday, okay? So the quiz will be available to you from 6 a.m. to 11:59 p.m. You can take it anytime during that period. Once you start the quiz, the timer does not stop, so you have to complete it, okay? But do come to class at 12:30. Okay, moving on. We have the cell membrane and nucleus. If we look at cells, we can categorize them into 2 groups. Okay? We have what's called the eukaryotic cells. Another name for it is eukaryotes. And then we have the prokaryotic cells, which we will get to in a second. Okay? We are made up of eukaryotic cells. Right? The cells are, compared to the other type, these cells are much more complex. They are generally larger than the prokaryotes. And the defining feature that separates the two is the fact that eukaryotic cells have a nucleus. And the nucleus is represented by like this orange circle inside the cell. Okay, so the nucleus contains genetic material. Our nucleus protects our genetic material, right? Our DNA, basically. Right? Prokaryotes also have genetic material, but they don't have a nucleus to protect them. In addition, eukaryotic cells have many types of organelles. Organelles are structures inside a cell with specialized functions. And we're going to learn about all these organelles. Right? So mitochondria is an organelle. It makes energy for the cell. Chloroplast is the organelle for plants. It does photosynthesis, right? So on and so forth. So each of these organelles have a specific function, and there are a lot of organelles in the eukaryotic cells. Animal cells, plant cells are examples of eukaryotes. Okay, so all the cells in your body are eukaryotic cells. Now, earlier we talked about the amoeba and the paramecium. They are not animal cells or plant cells, but they are also eukaryotes. Okay, also put down also like the amoeba and the paramecium, right? Okay, so they actually belongs to a group called protists. Okay, animal and plants are not the only living thing that exists. Okay, there are other kingdoms like protists, like fungus. Right, so they are all eukaryotes, eukaryotic cells. On the other hand, we have the prokaryotic cells, right, or prokaryotes. They are smaller, they are simpler in structure, and they do not have a nucleus. Do not have a nucleus. Right? They do have some organelles, but not a lot. Right? There is an organelle here, right? And most bacteria are prokaryotes. Okay. I don't know why I put the word most here. We can say bacteria are prokaryotes. Yeah. Does anybody know any bacteria? Any bacteria name that you read on the news? Anytime they have a recall?
E. coli.
E. coli, that's very good. That's exactly what I have in mind. Right? E. coli is one. Salmonella is another one, right? You might have heard of like Streptococcus, Anthrax. Right? All these are bacteria, and all of them are prokaryotes. Okay? They don't have a nucleus, and they are much smaller. Okay? Remember, they are measured in the scale of like 1 to 10 micrometer, whereas the eukaryotic cells are measured in the scale of 10 to 100 micrometer. Regardless of whether you are prokaryotic cell or eukaryotic cell, there is going to be a cell membrane that surrounds it. Another name for cell membrane is plasma membrane. And does anybody remember what is the major component of cell membrane?
Phospholipid.
Very good. Major component of the cell membrane is phospholipids. We learned that on Monday. And that's what these things are, phospholipids. There is a hydrophilic head, which means it binds the water. And then there are these hydrophobic tails. Okay? And together they form the cell membrane. In fact, there are two sheets of these phospholipids, and they form a selectively permeable barrier. Okay? Selectively permeable means only allow certain materials to pass through. Okay? You don't want everything to go through, right? You want to keep the harmful stuff out. You want to keep the nutrients in the cell. You don't want everything to just freely move in and out without any sort of regulation. Oh, by the way, like this hasn't happened to your class yet, but on the other day I lost connection during class, during like an online class. If I suddenly disappear, just stay put, okay? I will come back. Okay, don't go anywhere. Okay, so this is another view of the arrangement of the phospholipids by layer. You can see the outside of the cell and the inside of the cell, the cytoplasm here. They both are facing the hydrophilic portion of the membrane, and sandwiched between them are the hydrophobic tails. Okay. This is an actual picture of what the cell membrane looks like. It's from one of those high-power electron microscope. The black lines here, those are the hydrophilic heads, and then the white gap between the black lines, that's where you have the hydrophobic tails. Okay. Now you won't have to identify real pictures like these, but I'll give you a cartoon picture and ask you to identify the organelle. Right? If you need practice with those things, please do the study guide for lecture 3. Okay, so we're gonna switch gear and do a class notes. This is what we are drawing, okay? We are gonna be drawing this whole thing. So make sure you have enough space. I would recommend getting a piece of paper, turning it sideways, start here, and then draw it there here, and then we're gonna add some stuff here and some stuff here. Okay, this is plan ahead. All right. The reason I do the drawing is most people tell me learning the cells, if you just go through the slides and then you look at the organelles and it just like goes over your head, right? Like nothing really goes in. But if you draw it from scratch, if you build the cell bits by bit, it tends to help you understand a little bit more. Okay, so try your best to follow. Of course, I'll post the notes and post the video after. So if you need to see it again, you will have that option. Okay, so you will need some colors and you need a fresh piece of paper. Okay, this is if this is your paper, it's not something you can squish in the corner. Okay, this is a really big note. We'll keep on adding things to it as we progress. Okay. So what are we drawing? We are drawing an eukaryotic cell. I'll just give you a moment to get organized. Give me a thumbs up if you can when you have gather all your supplies, and then we're gonna do this. Okay, I got 4 thumbs ups. All right, let's get to it. So we will start with this guy. Okay, we start with this one. This circle, this sphere, this is going to be our nucleus, which is a defining feature of a eukaryotic cell. If I give you a cell and ask you whether it's eukaryote or prokaryote, the easiest way to tell is to see if they have this spherical structure in them, whether or not they have the nucleus. Okay. Now, the nucleus is actually surrounded by, give me a second, okay? It's actually surrounded by a membrane, okay? And we're gonna draw the membrane here for you. I'm gonna get a highlighter here. And I'm not gonna color the whole thing. I'm just gonna color a little bit of it, okay, like so. Like that. And I want to open it up so that we can see the inside. You guys know this chocolate. Here, let me show you a picture of it. You guys know this chocolate, right? The Ferrero Rocher. If you ever had one of those things, you can think of the nucleus like that, okay? The golden wrapper, that would be like the envelope that we are drawing, okay? It surrounds the entire thing, okay? Just a little bit. I love connecting things to food that we eat. It just makes so much sense to me. So here we have the nuclear envelope. Let's label that. And it surrounds the entire nucleus. Okay. Again, I didn't color the whole thing because I want to peel off the wrapper and see what's inside. Let me know if I'm going too fast, okay? Just pop me a message and I'll slow down. Now, on the nuclear envelope, there are all these openings. Right, so these openings, they are called nuclear pores. And the function of the nuclear pores are to allow materials to enter or leave the nucleus. Okay, some things needs to go in, some things got to come out. I'll tell you what they are in a bit. But you know, if the whole thing is wrapped, then there's no access, right? So by having a little bit of pores opening here and there, things can go in and out. Right? But it's not unregulated. Everything is like deliberate, right, in a cell. So there are ways to control what's allowed into the nucleus and what's not. Now, inside the nucleus, there is another circular structure. Okay. Back to the chocolate example. If you ever had one of those, you know in the middle there is like a nut, right? Okay, like a hazelnut. And that's kind of what this is like. There is a nut on the inside. There's another spherical structure, and this guy is called the nucleolus. Nucleolus. The nucleolus is responsible for it produces an other organelle. Remember, organelles are just these structures that we found inside a cell, and they have specialized function. So it produces another organelle called ribosomes. Not the blue I was looking for. Okay. We'll talk more about the ribosomes later. Right now, just know that that's what they are. That's where they came from, right? From the nucleolus. So back to the nucleus for a second. Right? One of the key functions of the nucleus is that it contains genetic material. And you should know our genetic material is DNA, right? But there are a lot of DNA. In each cell, the DNA, if you lay it all out, is going to be 2 meters long. And you're gonna pack it all into this tiny structure, this nucleus. Okay? It would be equivalent to packing 4 kilometers long of threads into a tennis ball. Right? So how do you fit it all in? Well, you're gonna actually have to package the DNA neatly so that they don't get tangled, right? And that they don't take up too much space. So I'm gonna do a little side description here. Right? So this is our DNA. Right, the double helix, we learned that last time, with the base pairs, ACTG, right? And we call this naked DNA. It's actually what it's called, okay? We usually don't find naked DNA lying around in a cell too often because if they are exposed, if they're naked, it's very vulnerable, right? They can get damaged, right? They might break apart. So often DNA will wound themselves around packaging proteins. Okay, so these are packaging proteins. Okay, it's called histones. And the DNA is basically going to go wrap around them like that. Okay, and it just keeps on going and going and, you know, forming a chain of them. All right, give me a second. So it's like I'm trying to find a nice picture here to show you. It's like a yo-yo, right? Okay, like a yo-yo. Right? The thread here is like the DNA, and then the yo-yo part of the yo-yo, I guess, that's the histone proteins. And you can take the string and you can wrap it around the yo-yo. Okay, except it's not just one yo-yo, many yo-yo, same rope. Once you run out of space, you continue on another one, and then another one, and eventually you get a bunch of DNA that are looped around these packaging proteins. Okay, and this whole thing that you get in the end, this is called chromatin. It's basically DNA wrapped around proteins, and that's the form of genetic material that you will find in the nucleus. Okay, chromatin. Does anybody have any questions so far? Is this clear? Okay.
So
Yeah.
all this is in the nucleus or in the nucleus.
In the nucleus, yeah, in the nucleus.
The nucleus,
Yeah, not in the nucleolus, right? Just the
not in the
nucleus.
nucleus.
Of course, you also have heard of chromosome. So how are chromosome different from chromatin? Well, it turns out chromosome and chromatids are essentially the same thing. But if you take this chromatin and you coil it up even further, if you give it more packaging proteins, then it's gonna, you know, form these really condensed network of DNA genetic material. Okay, and it forms like this really long coil fuzzy structure. Okay? And a lot of times, by the time it gets to this form, it would have made a copy of itself already, and then they would be stuck together at one point. And this whole thing that you see me drawing, that's the chromosome. Okay, so chromosome, if you search it up online, they usually show you a picture of an X, and that's like the most condensed version of the chromatin. Okay, we don't usually get the chromosome. Okay, in a cell that's not dividing, we have the genetic material in the form of chromatin. But when we divide, when the cell divides, the chromatin gets packed into the chromosome so that it's easier to move them around. Right? And like I said, before you divide, you need to copy all your genetic material, right? Which is why the chromosome typically will have like double stuck together. Okay, two of them stuck together. So are we all okay about regular DNA, chromatin, and chromosome? Any questions at all? Okay. So let's go back to our nucleus here. Now, I just want to give you another analogy, okay, in case you are still a little bit confused here. You don't have to draw this. You can just watch me do it. Okay? So let's say we have an avocado or something. Okay, avocado. This is the skin of the avocado, and then we're gonna color this guy. Okay, there you go. There you go. Okay, don't draw this. Okay, I'm just helping you understand it from another perspective. Let's say the whole avocado is analogous. It's an analogy to the nucleus. Okay, what would the seed be? What would the seed be?
Nucleolus.
That's right. The seed will be the nucleolus. And where will you find the chromatin? Well, you will find it in the meats, okay?
Yeah.
The part that you
yeah.
eat. And then the shell or the skin, I'm not sure what you call that thing, that would be like the nuclear envelope out here, right?
Yeah, with the pore.
Now, yeah, that's right. Now, if you take a fork or a toothpick and then you poke a bunch of holes on it, then you get your nuclear pores. Okay? So I hope that gives you a more visual way of thinking about the nucleus. Okay, try my best here. I hope you understand. Now we're gonna move on. All right, we're gonna draw more. The nuclear envelope actually not only does it surround the entire nucleus, but the nuclear envelope will extend into the cytoplasm to form a network of membrane. All right, so this is how I'm gonna draw it. Sorry, I'm just gonna move this guy a little bit to the left so I have some room. But here is the nuclear envelope, and then it continues to spread outward, okay, and form these network of membranes, okay? You can just like randomly draw some here and there like that. And then switch the color to something else. And you can draw a little bit more network, okay? Like that. So these are just an extension of the nuclear envelope into the cytoplasm. Just spreads out. All right, we'll come back to these network in a second, but I want to backtrack a little bit and talk about the ribosome here. Okay, so ribosomes, they are another kind of organelles, and we make them in the nucleolus. Once you create the ribosomes will leave the nucleus through the nuclear pores. And some ribosomes will be floating around in the cytoplasm on their own, okay? Just by themselves in the cytoplasm, like jellyfish in the ocean. Okay, so these are free-floating ribosome. Okay. Well, what do they do? Well, a couple of things. Number one, the ribosome themselves are made up of protein and RNA. And their function is actually to make more protein. Right? The ribosome produces proteins. Remember our previous discussion? Proteins are made up of amino acids. Right? So they're like beads that you connect together to make a long chain, right? So in the cell, that's the job of ribosome. They will find the amino acid and they will connect them together to build the protein for you. Now, I'm just gonna zoom in on one of these ribosomes, okay? I'm gonna zoom in on one of those things. They are really small compared to other organelles, but if we zoom in here, if we zoom in, and you have to believe me, okay? This is for real. I'm not making this up. You can read it in a textbook. The ribosomes is like a burger, okay? So there is a top bun and then there is a bottom bun. The top bun is called a large subunit, and then the bottom one is the small subunit. And the burgers are usually separated, but when they need to make proteins, they will come together and connect the amino acids together. Okay, so this whole thing, the ribosome, even though they're really small, but if you zoom in and look at them, it's actually two parts, a big part and a small part, and they will work together to produce proteins. Okay. Where do ribosomes come from? They come from the nucleolus. Okay? And they themselves are made up of protein and RNA.
Not DNA too.
Sorry, what's that?
Ribosomes are made of RNA protein, but not DNA.
That's right, not DNA. Yeah.
Not DNA.
Okay. While some ribosomes are free floating in the cytoplasm, some ribosomes will attach themselves onto this network. Okay, remember the network is an extension of the cell membrane, of the nuclear membrane. Okay, so some of them will have the ribosomes on them. So these ones that have the ribosome stuck on them, we have a name for them. Okay, this is called the rough. I'm really sorry about the long name, okay? I didn't make them up. That's just what they're called. Rough endoplasmic reticulum. RER. Okay. People look at that and look at these structures under the microscope and they look rough. They look bumpy because of the ribosome. Right? And on the other hand, we have the same kind of network, an extension of the nuclear envelope, but they do not have the ribosomes on them, so they appear to be smoother. We call them smooth endoplasmic reticulum. Okay, and they do different things. Can you believe it? It's already an hour. Right? Why don't we take a break? And when we come back, I will tell you all about these reticulums. See you in a bit. All right, let's keep going. So now that we have the distinction between the rough and the smooth ER, basically whether they have ribosomes on them or not, let's take a closer look at what each one of them actually do. Starting with the rough endoplasmic reticulum. Okay, so the rough ER actually together, the endoplasmic reticulum along with the ribosome, they produce and package proteins. into vesicle. Okay, into vesicle. Let me explain what that means to you. This is a little bit difficult to draw, but if I were to zoom in here, okay, zoom in. Whenever I do these little arrow things, we're zooming in, right? We will see that this is one of those networks, okay, and kind of stuck on the outside of the network would be the ribosome right here. Now, you should know by now, ribosomes will make the proteins. So these are the amino acids that are connecting together by the ribosome. Okay, and once you connect all of them, then the protein will get released into the inside of one of those networks. Okay. Perhaps it's best if you just watch the next part and try to understand it first, and then you can redraw it later if you want, okay? Also, you may choose not to draw this part. Okay? It's in the notes anyways, okay? Just watch. This is really hard to explain. Now, this protein, here, I'll put a label here. That's like a protein, okay? Freshly made by the ribosome. This protein is gonna move towards the edge. It's gonna move towards the edge, okay? And then I'm gonna have to draw a series of diagrams to help you understand this. As it moves towards the edge, it's gonna pull on the network. Sorry, give me a second. It's gonna pull on the network. Okay, so here's the protein edging towards the edge, and eventually, it is going to pull out so far that it causes a bit of the membrane to like bubble off, right? And ultimately, it will detach. Oops, wrong color. Ultimately, it will completely detach, and we have a vesicle with a protein in it. Okay. So this is what we call a vesicle. So again, you make the protein move towards the edge, and then it breaks off. The best way to think about this is blowing bubbles, right? Like, you know, soap bubble, right? Yeah, thank you, exactly, right? Blowing bubble. So if you blow bubble, the bubble goes like this, you know, you keep on blowing in it, and eventually the bubble comes off, right? It's exactly like that, okay? But instead of, you know, like an empty bubble, the inside actually contains proteins that was freshly made by the ribosome. And in the end, the name of the bubble is called a vesicle. I will post a video link for you later, okay? Probably not today because you guys are busy studying the quiz anyways. I'll post a video later on so that you can watch the whole thing. I don't like playing videos during classes. I think it's a waste of time, okay? But you can watch it on your own time. But any question about this process? Can you kind of visualize what I'm trying to describe here? Yeah, maybe. Okay, how about the smooth ER? What does that do? Well, to keep, can you explain the making of the protein part again? Oh, okay. You mean for the ribosome? Okay. So basically, protein, it's made up of amino acid, right? So I keep on saying you gotta connect the amino acids together. Okay? You take an amino acid, you need to form a bond to connect them together, right? So what actually makes that connection in the body? That's the job of the ribosome. Okay, so each of these red balls, red circles, each of them is an amino acid, and the ribosome will link all of them together. And once you form the entire thing, right? Once you form the entire protein like this, I told you it will fold up into like a ball, right? It'll fold up into a 3D shape. That's what I'm trying to draw here with this red, cluster. And then this will move towards the edge of the network, and then it bubbles off. Okay, hope that helps. Now, the smooth ER. What does the smooth ER do? The smooth ER is responsible for producing or produces lipids. Lipids. So anytime you need to make some steroids, you're gonna need smooth ER. Steroids like the sex hormone that we talked about, right? Testosterone, estrogen. Right? So you would expect to have more smooth ER in which organ, guys? Cells from which organ would you expect to have more smooth ER if we are talking about like sex hormones?
Yeah, testicle and ovary.
Exactly, right? So, and that is actually true. If you look at the ovarian cells and the cells from the testes, you will find more smooth ER compared to other cells in the body. Okay. The smooth ER will also help break down. Not only do they produce lipids, but they also break down lipids and drugs. Okay. A lot of people get this misconception every time. I realized this recently. People think when I say drugs, I mean like, illegal drugs or bad drugs. Not necessarily, okay? Medication that we take regularly, they need to be broken down. They need to be metabolized. And the smooth ER plays a role in that, okay? What do you guys take for headaches? Do you know the name of the medication? I'm sure you know, okay, but like tell me, what do you take for headaches?
Nanol.
Tylenol, right? Yeah, and if you read the Advil, right, these are the two main ones, right? Okay, yeah, ibuprofen, which is the active ingredient in Advil, very good. Okay, but for Tylenol, the active ingredient is acetaminophen. Thank you, Afra. And if you read the label on the Tylenol, it actually says, if you take too much, it may lead to liver damage. Okay. And so what happens is when you have drugs like that in the system, the liver cells actually will make more smooth ER so that it can help break down some of these drugs for you. Okay. And if you work those cells to full capacity, then some of these toxins might start to build up. So that's it for the drawing for the time being. Okay, don't put that away. Leave it next to you. We're gonna go through the PowerPoint slides, and I think we will have time to add a little bit more in about 15 minutes. Okay, so here we go. A lot of the information on the slides we've already covered. Okay, so I'm not gonna read them to you word by word. You should though, okay, when you're going through the notes again after class or whatever, whenever, read through the details information and match it with the diagram. Okay, so we were talking about the nucleus. We know it's surrounded by the nuclear envelope, and more importantly, we know it is going to extend outward to form a network, right? So this is like the professional drawings that you would get from like a textbook. You see the entire nucleus here. We see the nucleolus, and then the chromatin are going to be found here. What are all the yellow dots? Help me out here.
Ribosome.
That's right. Those are the ribosomes, and you get them from the nucleolus. Some ribosomes are floating around on their own. Others are stuck to the rough ER. If the ER does not have any ribosomes, then they are smooth ER. Like I said, it's everything we wrote down. This is another view of the chromosome and the chromatin and the DNA that I was telling you about. Right? We have the naked DNA right here, and they could wrap themselves around packaging proteins, and they could continue to do so to form the chromatin. This is the common state of the genetic material that you'll find in a cell. However, when it's going to divide, then you're going to convert the chromatin into chromosome. Again, these two things are basically the same stuff. It's just one is more compact than the other. The chromosome is more compact. The reason you want it to be more compact is during cell division, you need to move the genetic material around. Okay, remember we have 46, right? I gotta make sure when the cell divide, like you get 46 and you get 46. Right? In order to do that, I gotta move the chromosomes around. Okay, so I want them to be compact. It's easier to move things around when they're all compact. Think about you like moving to, like, let's say you move to a new place. You gotta pack your stuff into a suitcase, right? Carry that around. It's much easier than, you know, carrying your whole closet, right? It's the same logic here. You pack them up into chromosome, move them to where you need them to be, then you can unpack. You can change it back to chromatin later on. We talked about the nucleolus already. We talked about how it makes ribosome. We also talk about how ribosome, even though they look very small, but if you zoom in for real, they actually do look like a burger. There's a large subunit and a small subunit, and together they are responsible for protein synthesis. They make protein for you. The ribosome themselves are made up of proteins and RNA. This is an actual picture of what it looks like under the high power microscope. All these fuzzy dots in the background, those are the ribosomes. And these tracks that you are seeing, those are actually the rough ER. Okay, they are the membranes with the ribosomes stuttered on top of them. Okay, like sesame sprinkled on noodles. This is basically telling you that ribosomes can be on its own, free, or they might be attached. Okay, I've said that multiple times already. It should stay in your head now, right? Hopefully. All right, we did not discuss this yet, so I'm going to slow down a little bit and talk more about this central dogma of biology. Okay, dogma is just like a fancy word for theme. Okay, I don't understand why they can't just say theme. So central theme of biology is that we have all the information that's necessary for life locked up in the DNA. Okay. The DNA does many things, and one key aspect of DNA is that it contains instructions to make proteins. Okay. And DNA is really long. I told you already. Each cell has like 2 meters of DNA, and they're all crunched up in the nucleus. So it's impossible to get them out through the nuclear pores. And in fact, we don't want the DNA to be out and about in the cytoplasm because we might damage it. Right? We don't want to damage the DNA. If we damage the DNA, we change the instruction to make the proteins. We saw last class, it could lead to a bunch of problems, right? Like sickle cell anemia is caused by DNA mutation. Right? So there are other things that could happen when your DNA are mutated, okay? Like cancer, for example. So we want to keep the DNA inside the nucleus. But we want to make the protein. Right? So what do? We make a copy of the DNA in the form of RNA. Okay? So this process of copying a chunk of DNA is called transcription. To transcribe something is to copy something, right? If I ask you to transcribe the notes, you're copying the notes. Okay? You're not copying the whole 2 meters long of DNA, okay? You're just copying a section of the DNA. Right? There are hundreds and hundreds of proteins in the body. We're not making all of them all the time in every single cell. Okay? So you only need to copy the section of the DNA that will allow you to make a specific protein. Right? It's like this. Like, let's think of there's this like amazing cookbook in the library. Okay, there's only one copy of it. You're not allowed to sign it out. And so, but you really want to find out how to make the apple pie. Okay, there is a great recipe in there. So what do? You make a copy of the recipe, right? Not gonna photocopy the whole cookbook. You just copy the recipe that you need, and then you take it out of the library. You take it out of the nucleus, right? You bring it home, and then you use it. So this copy, this RNA copy, is going to exit through the pores into the cytoplasm. And the ribosome, the burger, is going to grab onto the mRNA and it's going to read the instruction on the copy. And it will use it to make the protein, just like you will read the instruction on the photocopy of the recipe to make the apple pie or whatever you're making. This process is called translation. Okay? Because DNA and RNA are the same language, they are both nucleotide. That's why it's called transcription. You're copying in the same language. RNA is nucleotide, but proteins are amino acid. It's a different language. That's why you are translating it, okay, from one language to another, almost like. Okay, so this is the central theme of how information flows in almost all life form. Okay, there are some instances that information might go backwards, but for the most part, we go from DNA to RNA to proteins. That's the central theme. Questions? Great. Oh, by the way, I got a comment. Attendance is still looking great. I'm still seeing 20 people coming to class each week. They are kind of, going down in my other sections. So I just want to give you guys some credit for being here. Okay, keep it up. All right, let's do another poll question right here. Oops, I pressed the wrong button. Hold on. Go ahead. Remember, if you don't see the poll, you can still participate by sending me your answers, okay? All right, let's end the poll here. Which of the following is a prokaryote? Prokaryotes are bacteria, so E. coli is an example of a prokaryote. The HeLa cells, they are cancer cells taken from cervical cancer, right, of the person. So it's from human, and human cells are eukaryotes. Okay. How about this one? If you have been drawing along with me, you should have no problem answering this question. Okay, I do want to keep this a little bit short because we still have quite a bit to do. Most people got it right. The correct answer is it produces ribosomes. Okay, that's the function of the nucleolus. And in case you forgot, the nucleolus is in the center of the nucleus, right? It's like the seed of the avocado, right? In our analogy. All right, so here we have the endomembrane system. In fact, our notes already cover a lot of these things already, but let's take a closer look. The endomembrane system is a network of membrane that forms tubes and sacs in the cytoplasm. Collectively, the entire endomembrane system works together to produce, to process, and to digest molecules like lipids and proteins, okay? Other large molecules might be involved as well. The system also recycles and get rid of some of the older molecules, or in some cases, the cell might be bringing in particles from its surrounding, and you need to deal with it. You need to break it down. That would also be the function of the endomembrane system. Now, it sounds confusing when you list it all out like that, but you know, let's relay it to our drawing. Hopefully, it will make more sense that way. These are the components of the endomembrane system. We already know about the nuclear envelope. We know the rough ER makes protein and package it into a vesicle. We know the smooth ER, they produce lipids and they help break down lipids as well as drugs. We have not learned about the Golgi and the lysosome yet.
I have a question.
Sure.
Yeah, so the endo membrane is related to the nuclear membrane.
Yeah, so the nuclear membrane, the nuclear envelope is part of the endomembrane system, right? And then the nuclear envelope extends into the cytoplasm and form other parts of the endomembrane system.
Okay.
Okay.
So yeah.
Sorry, go ahead. Yeah.
Because we have another membrane, the cell membrane.
That's right. So cell membrane. Yeah. So the word endo means within. Okay, within. Right, so the cell membrane is on the surface. It's like the barrier for the cell. But within the cell, there are a bunch of other membranes as well. And since they're inside the cell, we call them endomembrane. Okay.
Thank you.
Okay. So here we have the rough ER. We know the rough ER will work together with the ribosome to form the protein. And this is what I was trying to draw for you in your notes. Right? Maybe this is a little bit better. You have the ribosome, you have the protein. Protein will move towards the edge, and then it will bubble off. Okay, it will bubble off. So what's going to happen to this vesicle? Well, it turns out this is not the end of it. Okay, the protein that we just created, it's not quite ready yet. We gotta do some modifications to it. But before that, let's take a look at the smooth ER. Again, they are important for synthesis. That means producing various lipids, including steroid hormones. Right? We already talked about the ovaries and the testes having more smooth ER because they need to make the sex hormone. They will also help break down fats and detoxify drugs. So if liver cells are exposed to drugs, it could be regular medication that you're taking, antibiotics, that kind of stuff, then the liver will increase the amount of smooth ER and help break down these medications. They metabolize them. Okay. Next, we're going to talk about the Golgi body, and I think we're going to go back to our notes. We're going to draw a little bit more. And we're almost done with this thing, right? We just got to draw two more things, and then we'll be done. Also, part of the membrane system is this thing I'm drawing right now. It's called the Golgi body, and they are stacks of membrane, one on top of the other, kind of like pancakes. And we call this the Golgi body or Golgi apparatus. And I kind of want you to, like, when you're first studying for these things, don't try to memorize everything at once. Start like a little bit at a time, right? Start by memorizing key function of each organelle, just like one line function. Right? So for example, if I say rough ER, then you would say protein production and packaging. If I say smooth ER, they're like lipid production. Right? So for Golgi body, it will sort, modify, and repackage proteins. Think of them as the post office. Right? It receives all these things from all over the place, and they need to sort them so they know what goes where. Sometimes things that are arriving, the package might be damaged, then they have to repackage it, right, before shipping it off again, right? So that's what the Golgi body does. The vesicle is going to come. The vesicle that is freshly pinched off from the rough ER. They're gonna merge with the Golgi body. They will pass the stuff forward, and sometimes they might pass it backward, back and forth a couple of times. But eventually, they are going to create a brand new vesicle. Okay? And the content, the inside, is going to have a new modified protein. I'm just gonna choose like a different color here. Right? So this is a modified protein. Still inside a vesicle, but the contents are different. You don't have to know the kind of modifications, but in case you're curious, sometimes you need to cut the protein. Sometimes you need to add like tags to the protein so it knows where to go. Sometimes you might even need to add like sugar or lipids to the protein to make it into something that we'll talk about later. Okay, it's called an antigen. Okay, so there are lots of things you can do to the protein, and all these modifications, they take place in the Golgi body. All right, I'm going to zoom out a little bit, and I don't want you to forget we're actually talking about a cell here, so I am going to draw the cell membrane. Okay, so there's a cell membrane that goes around the cell. Okay, this is the nucleus of the cell. This is obviously not to scale. The nucleus is much bigger, but this here, this is the cell membrane. Okay, so just add a cell membrane somewhere in your picture. We're gonna need that for this next part. We're almost done with this note, okay? If you're running out of space, don't worry, almost there. Now that we have finished sorting, modifying, and repackaging the protein, like where is this gonna go? Well, there are a couple of places this can end up. Number one, it might move towards the cell membrane, and it actually will merge with the cell membrane, and it will release its content. It will release its content. Right? So that's one of the options. Protein get released out of the cell. Hey, maybe you need this right away, whatever this protein is. Yeah, maybe you need it in the bloodstream. So you shoot it out of the cell. Now it goes in the bloodstream, travels all over the body doing its thing. That's one option. Okay. Second option, stays in cytoplasm until it's needed. Okay. Hey, maybe you don't need it. Maybe you don't need it right away. Okay. We are just being prepared. Right? It's better to make it ahead of time while we have the resources, and then we'll store it in the cytoplasm. When you need it, we will have it ready. Okay, it's ready to ship. Right? You just say when, and we're gonna let it go. Okay, you might use it out of the cell, you might use it in the cell. It doesn't matter. We made it. It's ready. Number three, and I haven't told you this before, but I keep on saying that the cell membrane is primarily made up of phospholipids, right? We know there's a bit of cholesterol in it as well, but it actually, the cell membrane has quite a bit of protein in it as well. So maybe this protein that you make, it's gonna go to the cell membrane and become part of it. Right, so this is number three. Another possibility is the protein becomes part of the cell membrane. Okay. Are we good with these potential paths? Just talking about like what happens to the vesicle after it comes off from the Golgi. And then there's one more possible path. Number 4. This thing actually becomes another organelle. Called, I'm running out of color here. Let's use blue. Lysosome. Lysosome. Okay, I'm just gonna use a different color. There is the lysosome. Okay. All right, so we will end the lesson by talking about what lysosome does in the PowerPoint slide. You can add it onto the picture later, okay? But it's better to show you what it does. Okay, we did the Golgi already. It's like a bunch of pancakes, and these are vesicles from ER, right? It will go there, and then it will move back and forth, and then it will pinch off. Where do the proteins go from the Golgi? I show you 4 places it can go, right? So one of the places it could go is to become a lysosome. A lysosome. Lysosome contains digestive enzyme. We learned enzymes on Monday. These are proteins that speed up chemical reactions. And the lysosome actually has a lot of proteins in it, a lot of enzyme in it. It's like a mixture of enzymes in there. And all these enzymes help break down different kinds of macromolecules. And so, you know, if a cell ingested some kind of particle, I know it says food here, but like it's like they ingest some kind of particle. Okay, maybe it's a white blood cells that, you know, eaten up a bacterium or something like that. That needs to be broken down, and so it gets shipped to the lysosome, where they fuse together, and it gets digested. Okay. Similarly, if there is an organelle that's too old or too damaged, then it will fuse with the lysosome and it gets broken down. Okay? And then it will unlock some of the materials to be used again elsewhere. In some cases, the lysosomes will deliberately release its enzymes to kill the cell off. Okay? It does that on purpose. That process is called apoptosis, also known as programmed cell death. Sometimes this programmed cell death is part of normal development. When we were growing as a fetus, there are webs between our fingers. You can see the webs here, right? And of course, a baby is not born with webs between the fingers. So where do all the web cells go? Well, they die, right? The lysosomes in them release the enzymes and they kill those cells off. Apoptosis, programmed cell death. Okay? So that's part of normal development. Later on in the course, we will see there are instances where you do want to kill a cell off. Maybe it's dividing in a uncontrolled manner. So it's about to become cancerous. So you want to kill them off. You want to trigger programmed cell death before it becomes truly uncontrollable, right? Maybe a cell is not behaving the way it should. You want to kill them off, right? For the benefit of the entire organism, right? Okay, last slide here, and then we are done for the day. Like I said, there are a mixture of enzymes in the lysosome, and each of these enzymes, being proteins, The instruction to make them is in the DNA. And it's possible for the DNA to be mutated, and a person is born with missing enzymes or non-functional enzymes in the lysosome. Depending which enzymes are missing, the outcome might be different. Okay? Collectively, these diseases are called lysosomal storage disease. Most of these diseases are fatal in early childhood. I'll give you 2 examples. In Pompe disease, the enzyme that's necessary to break down glycogen in muscle cells are not present. It's not there. So the glycogen builds up and it interferes with normal muscle function, and it causes them to become weak. Okay, but not just like the moving muscle, but also the breathing muscle. And of course, the heart is also made out of muscle. So if the breathing muscles and the heart muscles becomes weak, you can imagine why it could be a problem. Okay. Tay-Sachs disease is another one. In fact, people can do genetic screening for this even before the baby is born. And they are missing a lipid digesting enzyme. And so there is going to be excess lipid buildup in the brain cells, interfering with brain development. Okay, so all those are related to missing enzymes in the lysosome. Okay, so just quickly here, ribosomes are made up of, we already wrote this down a couple times, it is the RNA and the protein. Okay, and the function of the ribosome is actually to make more proteins, right? Which of the following organelles are involved in breaking down macromolecules, toxins, and wastes? Okay. Lysosome does that. We just talked about how it has a lot of enzymes in it. But SMOOV-ER also does that, right? Detoxification of toxins. Your body views drugs as toxins because it's not like part of the body, right? It's something that's foreign, right? So it's gonna view it as a toxin and it needs to detoxify it, even though the drugs, the medication obviously is necessary for certain conditions. And that's it for today. Okay, we'll finish lecture 3 on when is it? Like Wednesday? Sorry, next Monday. And good luck. Study hard for the quiz. I am available on email if you have any questions before then. I won't be able to answer any questions on Monday though, okay? Because some people will already begin the quiz. Right? So Sunday is the last day you can send me questions regarding the quiz.
Can I ask you a question?
Sure, please go ahead.
What about the float